So our next speakers are Meghan and Michelle. And they have very long bios, so I'm going to let you read them, and I'm just going to summarize them very, very briefly. But it's so nice to be surrounded by such beautiful women. Look at you. Look at these girls. It's really nice to see all these powerful women in the room. That is wonderful. Not that we don't appreciate the powerful men in the room, but it is really nice to appreciate the powerful women. And so Meghan and Michelle are exactly that. They are brilliant minds that we've been working with now. Meghan joined our community 3 years ago. It was your first, was it 4 years? It was my first one, yes. Quincy was diagnosed. I came 4 years ago, and that's right. So Meghan has been a part of this, and she has seen the transition, and it's so nice for her to be able to come and speak to us about actually what they're doing and be able to share where they're going with their program. And so this is really exciting, and we're so honored and proud that Roche has joined the community as well. So Meghan is a PhD in biomedical sciences from the Department of Pharmacology at the University of California, San Diego. And in 2012, she joined Roche as a postdoc in the neuroscience group, working in stem cell biology and genetic neurodevelopmental disorders. In 2015, she joined the rare disease group at Roche as a discovery project leader, focusing on these neurodevelopmental disorders, and now she is heading that in Angelman Syndrome. Hold on. You're learning about Meghan through, yeah, you know, I am too. I knew that, Meghan. Michelle is actually a pediatrician. She's a translational medicine leader at the rare disease unit at Roche, where she led clinical development in rare disease development disorders. For early translational to clinical trials as a pediatrician, she specialized in disorders affecting the developing brain and understanding the impact on individual variation using genomics and imaging. Her medical training was in London at the Imperial College, and she also received her bachelor's degree in neuroscience with honors. Very nice. So it is very great to welcome both of them from Roche.
Thank you, Allison. That's a very kind introduction. So I just want to start by saying that I'm, of course, really happy to be back on the stage. As Allison said, I've been now here in the community for 4 years, and this has always been for me a really nice way to round off the year, to reflect on all of the progress we've made, that the community has made, and to get the momentum and the motivation to move even further in the next year. So with that, I'm here with my wonderful colleague Michelle, and we have a few topics that we want to cover today, starting with a brief recap and overview of where we are with our discovery program at Roche. And then secondly, to highlight an initiative that Roche started in collaboration with the AS community, in particular ABOM, to develop a concept model. And that has been in the works for the past couple of years and is now completed. And this will be published as soon as possible for the community to use moving forward in terms of developing new endpoints and talking to health authorities about how to set the regulatory path for clinical trials in Angelman syndrome. And finally, Michelle will talk to some new things that we're planning to do in next year around endpoint development, and that we're hoping will progress the field in terms of finding the right endpoints to use in these interventional studies. So just quickly, our disclaimer, basically the opinions and information that we express here are our own and not those of Roche. So in the rare disease group at Roche, we are highly focused on developing breakthrough treatments for individuals with either life-threatening or life-limiting rare diseases. But to do this, we really abide by 3 pillars. We start with the science. This is what kind of roots us in our hypothesis, what we think will work in terms of defining and finding transformative therapies for these debilitating diseases. And so with that, we start with the science and we follow the science. But that doesn't end there. We then, once we finally have a potential therapy, we need to also know how to bring it into the clinic. And with that, we need to have a good understanding of the disease. And that is a lot around knowing the natural history of the disease, but also being able to understand what is the unmet need and what are meaningful therapies for both patients and for caregivers. And to that end, we develop strong collaborations with you, with the community. And this is where we really hear the voice of the patient and get to know what it means to have Angelman syndrome. So I want to set the stage by kind of going back to basics in terms of the type of therapy that we're developing at Roche. The science has led us, and other companies as well, as you probably already know, to looking at a type of therapy which, broadly speaking, is an RNA therapy. And by this, this means that it's targeting RNAs. And so what are RNAs? So if we all remember that in our cells we have a genetic code, which is called DNA, and DNA is then used to transcribe RNA. And RNAs can be used to then either code for proteins, shown here on the lower left, or they can stay in the cell as RNAs and function as RNAs. Traditionally, drugs have often targeted proteins, but now in this new age of therapeutics, there are more and more treatments that are targeting RNAs, which are allowing us to very selectively modulate gene expression or gene activity. One example of an RNA therapeutic is an antisense oligonucleotide. And this is what you can see here on the right. And antisense oligonucleotides are small stretches of DNA which can bind to RNA and can act by either modulating the expression of the RNA or the splicing pattern, or basically the mechanism by which the RNA expresses the protein. And this has been demonstrated already clinically with Spinraza in spinal muscular atrophy, which you've heard about today. And there are other programs ongoing to develop RNA therapies for a number of debilitating diseases, including Angelman syndrome as well as Huntington's. So as you have already heard today and are very well aware, Angelman syndrome is due to a loss of UB3A in the brain. And this is due to a genetic defect on one copy of the UB3A allele that comes from the mom. Now, there's an endogenous RNA that is silencing the paternal UB3A copy, which is otherwise normal. By targeting that endogenous RNA with an antisense oligonucleotide, and in the case of the Roche molecules, this is an LNA antisense oligonucleotide, we can repress that RNA and allow for the expression of the normal copy of the UB3A gene from the paternal allele and replace the missing UB3A protein that is missing in the brain. This is the first step of drug development. So we have a strong scientific rationale and a hypothesis about how we can go about treating the underlying cause of Angelman syndrome. And in drug development, there is a stepwise approach to developing therapies and bringing them safely and effectively into the clinic to be tested in humans. So with this, we then move more into the later stage of discovery where we can test the molecules that we identify as potentially efficacious, but we have to also make sure that they have a significant safety window before we even consider to bring them into patients. And once we get into patients, start to move into patients, even at that point, there's a number of steps to slowly work our way into identifying whether or not the molecule is efficacious. So now to review where we are. So if you were here last year, you may remember that I presented this. So we started with a platform where we used patient cell lines, so stem cell lines, which were derived from patients with Angelman syndrome. And this was either from skin cells or blood cells. And we can use these cells to then differentiate them into neurons. And these neurons express the biological phenotype of an Angelman individual, meaning that they lack the expression of UB3A, as you can see by this blot, which compares a neurotypical control on the top to an Angelman cell line on the bottom. We can then use this cell line to screen thousands of LNA antisense oligonucleotides to identify the best molecules that upregulate UB3A protein to levels that are at typical levels. And from there, we usually end up with somewhere in the tens to hundreds of molecules that could potentially work. And then we need to go through a number of additional screening and efficacy measures in cells as well as in small animal models to test not only efficacy, but also some preliminary safety. From this, we usually end up with a small number of molecules, which we start to bring into larger animals, in this case, non-human primates or monkeys. And here there are a number of things that we look at demonstrating. First, and this is actually the new data that I'm sharing today that we've been working on over the past year. So first, we've tested several molecules now in monkeys where we've administered the molecule through a lumbar puncture, so intrathecal delivery. And we've been able to demonstrate that these molecules are able to reach the brain, that they distribute throughout the brain and reach the key brain tissues and are active in these brain tissues. And in particular, we see that UBE3A levels are increased to what we think are therapeutically relevant levels based on what we know from the work going on in the Angelman Syndrome animal models. And in addition to that, depending on the dose that we're able to use, we also show that we are able to get fairly lengthy mechanism of action in terms of the duration of action of the molecule, in terms of the level of increase in UB3A. So this is a very promising start as we move one step closer, but now we are in the process of rigorously testing these molecules still in the monkey to demonstrate that they have a significant safety window that we think is viable for bringing towards the clinic. So this is everything that we've been doing in the laboratory, but it doesn't stop there, because as soon as we get one of these potential therapies closer to the clinic, we also have to be understanding how we're going to bring that molecule through the clinic. And this requires a number of things to understand to allow us to have the best chance at success. And so this is what I've said before in some of the talks that I've given, but really highlights some of the key things that we're currently working on making sure that we understand as we move towards the clinic. And I think the community as a whole is putting a lot of effort into this. And this is based on, as I said in the beginning, having a deep understanding of the disease across the different age groups, as well as across different genetic subtypes. Importantly, we need to know what is the unmet need and what is clinically meaningful for patients and for caregivers, and I'll get a bit into that in a second with the concept model. We are working on understanding what would be the best outcome measures to use in a population, in the AS population, that is both robust and relevant to this population. We want to do our best to ensure that we design clinical trials in a patient and caregiver-friendly way to reduce burden. And finally, especially in rare diseases, it is very important to start to have early interactions with the health authorities to come to an understanding so that they're educated on the disease, and as well as that there starts to unfold a regulatory pathway for getting molecules or drugs to the market. So now I want to briefly go through and the highlights of the concept model, which we've been working on. At first, I want to just call out a couple people here. I want to first acknowledge Tom Wilgross from Roche, from the Patient-Centered Outcomes Research Group, as well as Terry Jo Bichelle from the Angelman community and ABOM, who really are the ones that have pioneered and pushed through with this effort. And they've done a fantastic job. The third persons that I want to significantly acknowledge are all of the people that participated in this study. So there are a number of caregivers who might be sitting here today, and I want to deeply thank you and acknowledge the contribution that you have made and how it will impact the progress for this community. So a concept model is really to help define the spectrum of the burden of the disease and to start to understand what are fit-for-purpose measures that we can use to demonstrate efficacy in drug studies. And this starts with first understanding what we know in the community already. So going through an in-depth literature review to basically say, this is what has previously been characterized. But beyond that, we then embark on a number of interviews with both clinicians, so expert clinicians, but most importantly with caregivers to get a broader scope of what does it mean to have Angelman syndrome. This is just an overview of the demographic of the people that were, or the Angelman individuals or the families that were interviewed. There was most of this was done in the U.S., but we tried to have an international outreach as well. Unfortunately, we were not able to extend this very far for the time being, so we were able to get 5 interviews in the Netherlands, but we are hoping that in the next efforts we may be able to make this a more global study. In particular, it is of importance to note that the caregivers that were interviewed spanned the Angelman individuals of the caregivers spanned the ages from 2 to over 18 years of age, so we have a good representation across different age groups. And in addition, we had representation of all the genetic subtypes. So this is a compilation of the information that comes out of hundreds of pages of transcripts, lots of data, and it is distilled down into a very simple message, and that is that these 6 things or concepts are really those things that were identified both by clinicians and as caregivers as the most important concepts to look at when we think about having a transformative treatment for individuals with Angelman syndrome. And these are very likely not to come as a shock to any of you. These include expressive communication, independence and self-care, improvement of sleep, the reduction of maladaptive behaviors, as well as improvement of fine and gross motor skills and reduction of seizures. All of these core concepts were represented across the different age groups. Some had higher representation in one particular age group over others, but importantly, they really are a core part of this disease and are reflected across the different age groups. And I think that this study in particular, while it, as I said, this might not be surprising to you, really gives us a validation of that and something that we can take forward in a robust way to communicate also with the health authorities. The last thing I want to just say is that this study also highlighted a number of things that were that have never been documented, that we may have heard from individuals. I for sure have heard some of these things before, but these are concepts that clearly came out as very important concepts to Angelman syndrome, but have not previously been defined as concepts in Angelman syndrome or impacts on the quality of life of Angelman individuals or caregivers. And this is an additional important part to this study and will help us also moving forward, because these are also things that we should be looking at when we start to conduct these studies, these drug interventional studies. So with that, I want to hand over now to Michelle, who will talk about what we're doing now that we have all of this information to start to help define the endpoints that we will use.
Thank you, Meghan. So I think the applause is really for all of you and the families who took part and contributed to this disease concept model. And it is a really important piece of work. But why is it important, and what are we going to do with it now? And this is really because any therapy for Angelman, not just ours, but anybody's, needs to address not only clinical symptoms, so what's measured in the clinic, but more importantly, the daily living needs of individuals with Angelman and the impact that that has on the families. And so this is where the disease concept model is helping us to guide our therapy development to actually address these needs. So how are we using the disease concept model now? So we've taken the concepts of interest, so the things that you have told us are the most important to you in your daily lives, and this includes improved expressive communication, independence and self-care, improved sleep, maladaptive behaviors, motor skills, and seizures. And Tom and our colleagues have mapped these concepts to the most widely used clinical scales that are commonly used in Angelman, so the Bayley, the Vineland, and the ABC. And what you can see here, first of all, is that none of these scales by themselves actually covers everything that is important to families. And the second aspect that you see is that there are actually some gaps that are not covered at all. So for example, sleep and seizures are really not covered by any of these scales. So what this tells us is that there is work to be done in developing clinical endpoints for trials for future therapies. And so an endpoint is the measures that we take during a clinical trial that will show us whether that therapy is actually helping those individuals in the study or not. And it's very important that we get that right, because we don't want to be in a situation where we might have a therapy that's working, but because what's being measured is not appropriate, that benefit is not being detected, and therefore families would not have access to that therapy. So that's why we really need to invest time and effort in getting this piece right before we move forward into the next stages. And so what are we going to do to bring that information together and get those insights? So we are planning an endpoint enabling study. So this is a new study that we are going to aim to start next year, which is a focused preparation for trials. This is a decentralized design, which means it will be focused in the home on the reality for people with Angelman and families. And it will involve new digital approaches, so new devices and wearables that we believe will bring a better insight into the reality and the potential effect of new therapies. This will involve, we will recruit 40 individuals, 40 children, 2 to 12-year-old with Angelman, and also 10 adults with Angelman. And alongside that, we will recruit 20 typically developing controls for comparison, because many of the things that we will be measuring in this study are new, and there is no comparison that we can go back to see how do Angelman children and adults compare to that group, and that's important for our discussions with regulators, the FDA. And we are planning to start this study in the beginning of next year. And really the primary objective is to develop new endpoints for Angelman. And so how will this study support drug development for the Angelman community? So the aims of this study are very specific, which is really to develop endpoints for trials, to use more realistic and quantitative, that means accurate measures, and to reduce the burden to families, because we understand how much of a commitment and a dedication it represents from the families who take part in these studies, and we don't take that lightly, neither for this study nor for any future clinical trials that we may run. So we want to really understand how we can minimize what is asked of families taking part in these studies. So as I mentioned, this is a decentralized design, so it will be focused on the home, which we hope will be more flexible and more acceptable to families. And this will be a focused and in-depth study of a small number of Angelman individuals over a set period of time, which is relatively short, so it'll be 1 year of duration for each individual. And we'll be looking at how new devices could be useful for clinical trials, and also addressing some of those gaps that I showed you earlier. So how EEG and sleep measured in the home could be used as biomarkers or endpoints in future clinical trials. And the main thing to take away from this is that really this study will be about developing endpoints for Angelman clinical trials. In the study, we will be using a number of wearable devices, and the idea behind this is to understand what is the most simple approach that we could then take forward into any future clinical trials. So we will be measuring a number of things together, and then the idea is to understand from that how we can simplify what we do next when we are able to bring a molecule, a therapy to clinic. So this is a particular type of observational study. That means it doesn't involve drug. So there is no drug involved here. And there are many different types of observational studies that can have different objectives. So this community is actually very fortunate that you have a number of ongoing observational studies, and it's really important that we gather this knowledge alongside developing molecules and therapies, so that when we do go to clinic, we're able to do things in the right way to really understand which therapies are useful for individuals with Angelman. So the focus of this study that I've just described to you is to really inform clinical trial design and to build a control cohort. So what we mean here is that we may be able to use these data, this information, which is being collected in a very rigorous and high standard way, which is very similar actually to how a clinical trial would really be later. And one of the things that we can do with this information is to use this as a comparison for future studies where we can compare individuals who are receiving a therapy with this information. And what this means is that we could potentially reduce the number of individuals that need to be on placebo in any future trials. We might be able to reduce the length of that placebo period, or if we are able to advocate for this with the FDA and other health authorities, we may be able to use these information instead of a placebo arm. So this is something that we will need to discuss with them and have agreed, but this could be something that would be really beneficial and of value to the community. And of course, as has been mentioned several times today, this can also help us to understand the safety of any new therapies. So I'm coming to the end of the talk, and really we've shared a lot of information with you in this time, and there are several clinical development building blocks that we've touched on. So Meghan actually started by sharing with you some exciting new data from our work in developing a new therapy for Angelman with our LNA. She also shared the disease concept model and the important results that you have directly contributed to, and how we're going to use that going forward. Megan Cross has been talking about the global registry, and that is a really important effort that I really want to also shout out and encourage families to participate in, because it's another key building block of clinical development for trials and will become increasingly important. I then shared with you the information about the Endpoint study that we are going to take forward next year, and also about the importance of continuing to contribute to these studies. And we are aware that this is a significant commitment from families, and that there is a lot going on in your lives already, and that this is an extra request. So it's about understanding that this will have a direct value for actually running clinical trials the way that they should be run, to really reflect what's needed. And then the final important piece is really a collaborative approach, which is that we need to work together. And so this Endpoint study that we are designing is in collaboration with Biogen and Ionis, who we are working with to be able to share the data from that study so that it can bring the biggest value and benefit to the community. And we will also be working to share those data with the FDA-funded natural history study so that it doesn't represent a conflict for families regarding which study to take part in, and so that there will not be duplication of assessments. So that, again, it's the most common sense approach for families. And so really just to close, I want again to thank all the families who have participated and who continue to participate and contribute to these studies, and that these are really the steps to take us forward into clinic, and we look forward to making that step together with you very soon. So thank you very much.
questions before the break? I promise you'll get a break this time. Any questions? Did I see a hand raised? Yes. While we're waiting for the mic, the other thing I just wanted to announce, and I have it on my email, is that there's also a booth outside where you can visit that is looking at a new natural history study that's being done at Boston Children's Hospital that is NIH funded, and that is going to be starting in 2019 as well. Again, more information, another request of families, but I do ask you to go out there and talk to them and talk about what they're looking for in terms of enrollment and participation. So there's a lot that we're requesting of you, but the good news is that we're putting all this work forward now because we're so close to needing to look at this information for clinical trials, which we wouldn't have been able to say to you 3 years ago. So this is very, very exciting. Yes.
Okay. If your child is going to be in one of the Ovid trials, could they also be in yours because they will be some of the same type instruments being used?
That's an excellent question, thank you. So we would allow a child who's in another observational study, so a child that is not actually receiving a therapy through another investigational study, to take part in this. But if they are in the Ovid trial, that means that they are in another investigational study, so they would not be able to take part in this trial, because the hope is that that therapy would actually be having an impact, which would hopefully be beneficial on that child, so or that individual. So if they were receiving that therapy and it was helping them, it would be changing the way that they are developing and progressing, and so we would not then be able to use that information for the purposes of it being a placebo comparator in future. So that would be the reason why. I appreciate that it would then mean that we couldn't use the data across, but it would be for a good reason.
Any other questions? I have a question that I expect a lot of parents will want to know. So a lot of children, particularly because you're looking at a large group in the pediatric population, a lot of parents change medications and supplements and things, foods, diets. What is going to be your exclusion criteria in terms of people starting CBD? People, and is there just they can be on what they're on, but for a 3-month duration and nothing can change? What's going to be your inclusion and exclusion in that regard? Because that's pretty common particularly in the youngers.
So in this setting, we want to actually learn about the reality of what's happening in daily life, and so we would ask that if people have a sense that things are going to be stable, or that if changes want to be made, that maybe, as you mentioned, that those changes could be made maybe like before they join the study, so that if you do want to start a new therapy, whether it's whether it's CBD or whether it's speech and language therapy, or these interventions that we do really feel can be very beneficial and can really support any other therapies, that is done in advance of joining this, so that can stabilize. We wouldn't exclude people. We wouldn't say that don't do something that's beneficial for your child in order to join this study, not at all. But we would just ask that maybe if that could be stable when you join, that would be the best. But we also recognize that things change and that unexpected things happen. So if there's a need to start a new medication, or there's a recommendation from your doctor that something should be started, that we would just ask that people are very open with us about that, so that we know. There's not going to be any repercussions from that. It's just about us knowing documenting what's been happening, so we can understand that, like she got a lot better and it was because they started a new therapy and that's why. So we can understand.
Thank you.